Thank you for joining us today. My name is David Su. I'm CEO and co-founder of Kamada. We are very excited to be hosting today this panel discussion, which brings together renowned experts, clinicians from North America and Europe, all of them with a lot of experience treating Alpha1 patients. Kamada is committed to innovation and novel treatment for the benefit of patient population. Our Inhaled program is well aligned with this strategy. And we believe that this might be a breakthrough therapy for the benefit of the patient population. I would like to introduce Pnina Strauss, our VP Clinical Trials and IP. Pnina will be introducing today the panel members and will present part of the trial results. Pnina, please.
Thank you, David. Hello, everyone, and thank you again for coming. We are very happy to be here and excited with the update of the results that we have. So let's start. So this is a forward-looking statement. We have to show you the slide. You're welcome to read it. By the way, all the presentation will be available for the public in the Kamada website after this session. Not sure if immediately after, but it will be in the Kamada website very soon. So we are a public company. It's important to note that everything that I'm going to say or anyone else is not a recommendation or a non-recommendation to handle or sell our stock or buy. So a must slide. Introduction, panel members and chairman. So we have program chair, Professor Robert Sandy Sandhaus, professor of medicine from the National Jewish here in Denver. From the panel members, we have Professor Kenneth Chapman, Director of the Canadian Registry, and working as an investigator in the University of Toronto, again, for pulmonary medicine. We have Professor Jerry McLevaney from the Beaumont Hospital in Ireland. We have Professor Robert Stockley from the Birmingham University in the UK. And we have Dr. Jan Stolk from the Department of Pulmonology in Leiden, the Netherlands. Okay. So just a bit about our study before we share with you the results. The indication of the study is Alpha1 deficiency patients who have congenital deficiency of Alpha1 and emphysema. The investigational product that was in the study was Alpha1 Antitrypsin from a plasma-derived source using an inhalation device called the eFlow from PariPharma GmbH in Germany. The study drugs was also the placebo that comprised the buffer of the investigational product, the non-active ingredient of the AAT preparation. In terms of study design, this was a phase 2-3 double-blind, placebo-controlled of 168 subjects. We had a 50-week treatment of a double-blind period and then an open-label extension for another 50-week. The trial was designed approximately 8 years ago together with the EMEA and with key opinion leaders worldwide, European and US. The sites in the study, as you can see, most of the sites were in the Western European countries and in Canada. The study was also comprising of a DSMB committee that was in the US, Spain, and Italy. So what's new, what's innovative in this study, the primacy of the study? So first of all, this is the largest study that an inhaled AAT was ever applied. Second, the use of e-diary. In this study, each patient had used an electronic diary card on a daily basis, answering daily questions on their symptoms, on their safety, adverse events, changes in their medical history, changes in their feeling, and this data was collected for a period of 1 year. So we have a lot of data collected on the natural history of the disease of these patients on a daily basis, and we are very happy to note that the amount of information that was collected from the study is able to make a difference in terms of the information that we have today about the alpha-1 deficiency. In terms of efficacy, this is the first ever controlled randomized trial that was able to demonstrate lung function efficacy. There were a lot of follow-up trials, registry trials, open label trials, but this is indeed the first placebo-controlled randomized trial that is showing difference in lung function. Inclusion and exclusion, as you can see, these are quite standard. These are the main inclusion and exclusion criteria that were used to select the patients in the study. Of course, alpha-1 deficiency, the FEV1 had to be less than 70, and at least they had to have at least 2 exacerbations in the last 18 years from baseline, from screening. Most of the patients in the study were naive. We had 2 sites in Germany that were also enrolling patients who were on augmentation therapy. This was the direction of the EMEA. In terms of exclusion, again, quite standard exclusion criteria for a plasma product, for example, IgA deficiency, also smoking, and again, anaphylactic reactions to human plasma-derived products. Study endpoints. The primary endpoint for the study was the time to first exacerbation of a severity of moderate to severe based on the event-based definition. The secondary endpoints were time to the first event-based exacerbation, this time mild, moderate, or severe. Additionally, we had the severity of the first exacerbation of the study, and we had also rate of exacerbations in the study. The safety profile here included the adverse event, lung function, vital signs, and again, the standard physical exam, EKG, and lab evaluations. In terms of the regulatory guidance that we received for this study, although the guidance at the time that we designed the study was still a draft, indicating the applicability of the primary endpoint, the EMEA was saying that they would be interested also looking at the severity of exacerbations, at the rate of exacerbation, and most of all, they will be interested to look at the totality of the data. The terms totality of the data were used constantly throughout the term of the study when we approached for scientific advice, and again, just recently when we approached the EMEA, and I'll discuss this in a minute. So what's new? We have already issued a press release about the results, and we are bringing now a new update. So the results that we have released in the press release indicate that the primary endpoint was not met. With that said, additional analysis indicated that lung function analysis and first exacerbation severity did show statistical significant changes in favor of the AAT group. We have approached the EMEA and presented the data and asked for additional guidance. The EMEA confirmed that this orphan designated drug, will be reviewed also based on a post hoc analysis, as well as review again of the totality of the data, irrespective of the fact that the primary endpoint was not met, meaning that they asked additional information, additional statistical methods that we would apply, and they said, bring us the data and we would look at the drug again. But the bottom line is that they didn't rule out the study, and they are willing to review it for submission based on post hoc analysis. The analysis strategy, due to that, is based now on lung function as primary analysis, exacerbation symptoms as complementary or secondary analysis, and of course the safety of the drug. So going to the results. This table presents the baseline characteristics. As you can see, the table mostly shows that the two groups of AAT and placebo were quite harmonized from the start, except for three main parameters: age, BMI, and oxygen users. In the AAT group, we had more patients who were a bit older, older than 60, more patients who had BMI which is lower than 20, that may potentially correspond with a more severe form of a COPD disease, and more oxygen users, again, that may correspond with a more severe form of COPD. Spirometry measures. Here you have a graph of the FEV1. Blue is the AAT group, red is the placebo group. The overall effect is presented here in the AAT group as change in 50 mLs, and the overall effect of the placebo group in minus 27 mLs. The p-value for this overall effect corresponds to 0.0268. This was done by a method called MMRM. This is a method that the EMEA requested that we apply. It's called the mixed model repeated measures. And the overall effect essentially represents the, as if you call it, the area under the curve, the entire effect from day 0 to week 50. Same graph here for the FEV1 versus SVC under the method of the MMRM. Overall effect for the AAT is plus 0.62. Overall effect for the placebo group is minus 0.87. And the p-value is 0.0074. This table summarizes the changes in lung function. Here you have three parameters of lung function: FEV1 in liters, FEV1 presentive predicted, and FEV1 of SVC. You have two tables for these three parameters: changes at week 50 from baseline and the overall effect, the graphs that I've shown you. So you can see that the changes for both changes at week 50 and the overall effect are either significant or close to significance. This table is about the diffusing capacity, the DLCO. The bottom line is that we didn't observe any changes in the diffusing capacity in between the groups. The nature of the first exacerbation. Just before I give you the result, just to remind everyone, the Antonisen criteria, the symptom-based criteria, call for type 1, 2, and 3 categories that are basing this categorization on three cardinal symptoms: dyspnea, sputum volume, and sputum color. Type 1 is having all three major symptoms. Type 2 is 2 of the 3, and type 3 is 1 of the 3 major symptoms. Beneath the table you have a scale, and you can see that the dyspnea is considered to be the most cardinal of the two in terms of the severity of the exacerbation of the event, and the sputum volume and sputum color have a different, a lower scale. The nature of the first exacerbation, when we look at the Antonisen type 1 to 3 criteria, we can see that for type 1, in the AAT group versus placebo, we have a lower number of events that are categorized as type 1, in which they suffer from the 3 cardinal symptoms. We do see a shift potentially in type 2, where the AAT group has more events of type 2 versus placebo. But our main focus is the fact that potentially there is a change here. The AAT is able to make a change in the nature of the exacerbation by changing the severity of the exacerbation from the most severe form of type 1 and potentially have more events of type 2. When we break down the Antonisen criteria, the symptom-based definition into symptoms, dyspnea, sputum volume, and sputum color, and look at all exacerbations, type 1, 2, and 3, for either 10 and 14 days, we see that the AAT group versus placebo is improving for dyspnea and for sputum volume, including having a statistically significant signal. For sputum color, we didn't see the same effect, and actually this is for the benefit of the placebo group. So the bottom line here for this slide is that during first exacerbation, the AAT group improves significantly, as you can see, for the dyspnea and the sputum volume symptoms. This graph represents the continuous evaluation of the dyspnea score. And the next graph that I'll just turn is the well-being score. I'm going back now to the dyspnea. So you can see that the two graphs are very much alike in terms of their shape. The AAT starts higher in a much worse situation and improves, while the placebo continues to be the same or escalates a bit. And this corresponds quite nicely with the well-being score that correlates with the dyspnea, being the most severe form of symptom that patients suffer from during exacerbation. It's important to note that these changes were not statistically significant, but only observed as trends. From a safety perspective, when we look at the frequency of AE per patient per month, we see that the frequency is quite similar between the two groups. There were no AEs that were indicating immunogenicity or bronchospasms. There were no anaphylactic reactions. There was no pattern in the AE in any group, and the nature of the AEs was similar between the two groups. Just an illustrative picture of the strengths versus constraints of these results. So in terms of constraints, we do acknowledge the fact that there's no improvement in the primary endpoint at the time to the first moderate or severe exacerbation, and there's no improvement in the rate of exacerbation. With that said, we do realize there is spirometry efficacy. We do see an effect on the exacerbation nature. The drug continues to demonstrate its safety and tolerability. There is primacy here in terms of the clinical efficacy, as was indicated in the start of this presentation, and the unmet patient need exists. So in summary of the results presentation, we see efficacy in lung function, which is statistically significant. We see a change in the nature of exacerbation. We see a reduction in the type 1 in favor of dyspnea and also sputum volume. The drug is safe and tolerable. This is an orphan designated drug, and the drug is coming to provide an answer to an unmet patient need in terms of clinical primacy and efficacy data and AAT deficiency in general. So what's next? Moving forward, for the EU front, we are about to compile the MAA, the Marketing Approval Application dossier to the EMEA, and we expect submission by end of this year. In terms of the US front, we plan to approach the FDA with the results and obtain guidance on the clinical and the regulatory pathway in order to continue for the licensing strategy in the US. I think it's very much important to say that we remain highly committed to the Alpha1 patient community and bring the inhaled product into the market to provide an adequate and a safe, efficacious answer to patients and to this unmet medical need of these orphan patients. Special thanks, I will not read the entire list, but special thanks to our study investigators, to our medical advisor, Pablo Fernandez, to our DSMB members, of course to our patients in the study, to the entire teams that were helping us to generate the results, statisticians, coordinators. Special thanks to the AIR group who supported us in consultation, advice, and the excellence centers that performed the study, and of course to the Kamada team. I would like to invite Dr. Sandhaus to chair the panel based on the results that I've just introduced.
Thank you. Thanks very much. Thanks for this interesting presentation. We've all been waiting to see what the additional results would be after hearing the one piece of disappointing news that the study hadn't met its primary endpoint. And I think this is an intriguing addition to the information that we've had before us. I want to mention that I was not an investigator in the study. That's why they made me the moderator, because I don't know that much about it. And but on the positive side, we pretty much have a panel made up of the major clinician investigators in the world right now in Alpha-1, the major publishers in terms of clinical effects of Alpha-1 Antitrypsin Deficiency. And so I think we can have a rousing discussion, which I'll start by asking some questions of the panel, and then after I've gone through those, we'll throw the discussion open to the to questions from the anyone here that would like to ask. I'll use a baseball analogy in the hopes that the Israeli people here won't understand it, and that is, you know, my questions are relative softballs, and you guys can throw the hard balls. So the first and obvious issue that comes up in my mind is the results that we've seen for pulmonary function testing. Really, that's been a problem in previous studies that have been done demonstrating a change in lung function testing, the most simple test that we have to evaluate progression of alpha-1 antitrypsin deficiency. And we finally have a study that's giving some tantalizing results regarding lung function. And so I'd like to ask the panel to comment on that and put this in some context. Ken?
The changes in FEV1, do we have a microphone turned on? Just good. The changes in FEV1 are striking, and striking in a couple of senses. One, the numerical change is large enough to be clinically important. Two, it was accompanied by symptomatic changes. Patients during those first exacerbations felt less dyspnea, and the character of the exacerbation changed somewhat. Going back to what the event was, the graph that Nina showed us was of a relatively rapid change in FEV1 occurring soon after the onset of inhaled alpha-1 antitrypsin treatment, suggesting not a preservation of parenchyma, indeed there were no changes in DCO, but instead an airway event. And we can speculate, and maybe I should underscore speculate, that the airway event was a very important anti-inflammatory event. Just going back to the inclusion criteria, these were exacerbation-prone patients, at least 2 exacerbations in the preceding 18 months. So there was a very rapid and numerically important, symptomatically important change in lung function, and something that appeared to be quite distinct from what intravenous augmentation therapy might do.
Anyone else want to comment?
Right. Well, Luciana and I were very heavily involved in the design of this study right at the very beginning. And physiology was just really put in as a safety thing because, as has already been intimated, the studies that have been done with intravenous therapy have shown very little efficacy in terms of lung function. So when we saw the results, the first thing was, goodness gracious, why did that happen? And I think that we spent a lot of time thinking about this. And the first thing we'd have to say is, well, this is a slightly different patient population, because it was enriched for exacerbations, because that was the part of the outcome we were most going for at that stage. But it does look exactly like an anti-inflammatory response that one sees. And so I've been involved in a few studies of drugs that have anti-inflammatory effects. So we would know that, for instance, roflumilast will produce this same sort of improvement in FEV1 in a placebo-controlled trial. And also, when we did a study with an AZ compound in bronchiectasis, again, unexpectedly, we saw almost exactly the same rise in FEV1. So this, you could argue, is a bit out of left field, but if you start and sit and think about it and knowing the function of alpha-1 and the delivery of alpha-1 and the patients that we looked at, then there's an explanation for it, and it goes along with what we would expect to see.
Right. I mean, we have investigators in you and Mark Brantley in the audience who've already demonstrated the anti-inflammatory properties of inhaled and intravenous alpha-1. So this is an interesting finding for sure. There's even the suggestion that the rate of decline of FEV1 that we're seeing in the treated group is roughly equivalent to what we see in normal individuals. Do you think we can actually go that far in suggesting that?
You can go as far as you like, I guess, is my answer to that. But, I mean, I think you have to look at the data in its totality. And, you know, as I said, this was a bit unexpected, so we've been looking at it and we've been thinking about it. So you're right, by the end of year one, and unfortunately, by design, it was a one-year study, and of course, we'd all like to now say, well, let's see what would happen over 3, for instance. But by the end of year one, there was this difference. Now, does that just reflect this kick-up, and that's all it was? And I think it's probably more than that, because if you look at the kick-up and then you watch the rest of the slope, it is still diverging. But it makes it very difficult to put exact numbers on what it means in 1 year, when we're still trying to understand, firstly, why it went up, and now, what is the real course, and what have we affected? I mean, just to mention the gas transfer, I think interpreting gas transfer is not easy. I think people think it's easy. And one of the problems here is that we've opened up airways, and that's probably why the FEV1's gone down. Now, if you open up airways that have been supplying emphysematous areas, the KCO could actually go down, the DLCO could go down, but it could go the other way, because you open up bad airways and your KCO may get worse. So interpreting it's very, very difficult. What we're pretty sure is we've done something to the airways. And that now has potential ramifications for long-term progression of lots of things.
And what we're pretty sure of is that we're confident about the DLCOs that are done in each of our institutions, but when you group them together, we've never really been able to show a DLCO signal in any of them.
I'm any confident of mine. I think it can be done very well, but you have to have very, very good QC. And if you do good QC, it is actually more sensitive to change than the FEV1. The FEV1, of course, is very much more robust. But when you see the data, you have to ask, why has it gone up and why has it gone down? And just because it's gone up or gone down doesn't mean to say you've made emphysema better or worse. So you have to interpret it with the totality of the data, including alveolar ventilation itself.
So I want to ask Jan about when you look at the overall picture that was just presented, how would you apply that clinically to patients that you've been seeing? Where do you think that effect fits in with what you're seeing?
Yeah, well, right in my clinic, I would like to confine to those patients that were actually eligible for this trial, and then really try to understand whether we have been treated so-called small airways, or maybe a little higher up in the airway tree. That would be interesting to know. And I'd like to add to the data that in the protocol, it was mentioned that all patients should be measured 4 weeks after stopping the inhalation treatment, and then the data show that there is a return to baseline of the FEV1. So the hockey stick phenomenon in the first 4 weeks also occurs at the end of the study after stopping the treatment. So that alludes you to, I guess, airways disease components that have been treated, and I think those are relevant for my patients. Especially in my country, because there's no IV treatment.
And so, Jerry, why do you think that we haven't been able to show spirometry changes, for instance, in the IV trials that have gone on so far?
Yeah, so that's a really interesting question. I would think what we are seeing here is a direct anti-inflammatory effect on airways. And that's why we're seeing an early effect, and that's why we're seeing an increased FEV1. And it's partly due to the fact that when you give an aerosolized form of alpha-1, you're getting very high levels of alpha-1 antitrypsin on the epithelial surface. much higher than you get with intravenous alpha-1 antitrypsin. And I think that's the key thing here, that you're seeing a real airway effect because you're getting more alpha-1 to where the inflammation is. You may not see a DLCO effect for other reasons. The question for us is, not only is this applicable to alpha-1 antitrypsin deficiency, but other inflammatory airways diseases, and I think it does open up that whole question. But the rapid improvement in FEV1, the sustained improvement in FEV1, that suggests to me you're targeting the inflammation in the airways, which is either directly by inhibiting neutrophil elastase or the downstream effect of neutrophil elastase. And that's what I think we're seeing.
Well, to kind of change direction, I've certainly participated in a number of studies that have had diary books and retrospective questions of patients about their symptoms when they come in for, the occasional visit. How do you think the e-diary worked in this situation?
Oh, it was fantastic.
Did you design it?
Correct. People have used diary cards for COPD and exacerbation studies, and it's like anything. If you get a patient to fill in a diary card, you're never quite sure when it was filled in. And they could be doing this in the car park and all sorts of things and go on.
Absolutely.
So here was an e-diary, and I'm tempted to say, if they didn't fill it in during the day, they got an electric shock. But they didn't. What was done was they were reminded they hadn't filled it in the day. Now, even so, there were some patients who just lapsed, and so we accept that. But the vast majority didn't lapse. What this now meant is you had real-time, day-to-day symptomatology, which is unique. And looking through it, it's changed my concept of what an exacerbation is, because in the definition that Gold uses, for instance, they say it's an acute event. And sometimes it is an acute event. And sometimes it just creeps on and then they suddenly become unwell. Mm-hmm. Sometimes it just goes in bursts and then they get an exacerbation. So what happens to patients now is starting to become real to us.
Did it pick up exacerbations that would have been missed otherwise?
Oh, absolutely. So again, people will know that there's this terminology of reported or unreported or treated or untreated, basically being the same things. It's an event where the symptoms change and the patient doesn't request treatment. So those events are in there, and they have been documented, and as you've seen from what Penina presented, we're actually starting to break down the nature of those episodes. And I would go right back to the very beginning. I didn't think time to first exacerbation was the outcome. Now we were constrained because that's what people understand and what the EMA said. But knowing about the inflammation in the airways of these people, with and without alpha-1 antitrypsin deficiency, it was reasonable to assume, I felt, that what we would do was change the nature, not necessarily the time. And I think that that's what's coming out of this. And so there's more analysis to do, but I want to say, well, are the unreported events, the so-called milder events, the patient's not quite sick enough to want to go on and take some treatment because, antibiotics might be a bad thing, so they sit tight a bit longer. And have we, by reducing the inflammation in the airways, made the threshold of those events now much, much higher, so they don't report them so much, and then when they report them, the nature has changed and their treatment has changed and the length is shortened. And that would be the real analysis from my point of view.
So this e-diary is available on iTunes now?
No. But it has been validated on the Palm Pilot, and I think once we understand this, there's no reason why that shouldn't be. Well, it's which included the electronic color chart. Well, it wasn't electronic; it was just
for the speed and color.
Yeah. So they were filling in those symptoms every day at the same time, and not just the ones here. There are other symptoms that we have filled in. And so we were able to look at all events, what their nature is, and I must say that having gone through 48,000 lines of diary card on at least 3 occasions, I think we could give you the job of doing the rest.
Thank you. Well, let me ask Ken about the elephant in the room. Why didn't the study meet its primary endpoint?
I would agree with the allusion that Rob made that it was perhaps the wrong endpoint, a very ambitious endpoint, and one that was likely chosen or negotiated with regulatory authorities because it's a familiar endpoint. But bottom line, the familiar phrase comes to mind: size matters. In this case, the size of the study. If we study exacerbations in alpha COPD, we expect to mount a study of 1,000 or 2,000 or 3,000 patients. This was a study of under 200 patients. It may be the largest study of inhaled alpha-1 antitrypsin ever done, but that's still a small study, and I think numerically too small to show the sorts of changes that we traditionally do in larger studies. So I think it's fascinating that we see a change in the type of exacerbation, and maybe that's a hint, but I'm not altogether surprised that we failed the primary.
Right. I'll make my own editorial comment. There are two aspects to the exacerbation endpoint. One is the fact that it's well known that when you enroll patients in an exacerbation study on the basis of them having frequent exacerbations, that the exacerbation rate always goes down in people once they enter a study. And the second thing is that this is pure editorial comment on my part, there is this mistaken impression, I think, that alpha-1 patients necessarily get a reduction in their exacerbations, because at least in the patients where augmentation therapy is available, lots of other things change in that patient when they start augmentation therapy. They get a nurse that visits them every week, that asks them how they are, that starts the IV, that says, oh, you're having a little cough, let me refer you to your doctor, or something like that. And so there are a lot of confounding factors around an exacerbation endpoint in alpha-1. But there were some other aspects to the exacerbation analysis that were very promising, like the dyspnea scores and things like that, which really seem to correlate very well with how someone feels they're doing. And what do you think about those kind of changes that we're seeing?
Okay. Yeah, I think all the credits have to go to Ed Campbell and Rob Stockley for designing this methodology, because at first view it looks really simple. There's the simple scores that patients have to fill in, but it's actually the analysis that is the hard part. It's not only reading 48,000 lines, but it's also trying to interpret the data coming out of that. And so as that has been done now partially and not completely, it looks like the symptoms which are most important to the patients, I think, like dyspnea, is going along in the same direction as the spirometry data. And I think that is an important message for my patients.
Great. Well, one of the things that stood out in the demographics that were presented today was that the treated group seems to have have some differences from the placebo group in spite of the randomization, and that those differences seem to be in the direction of the treated group being more severe in terms of some of the measurements that were done. Is there any feeling about how that might have changed the results or affected the results, or moderated the results, I guess?
Well, I'll start. You try and match in a trial, because that's important. And just by chance, this didn't match, and by chance, sometimes it won't. But, having treated alpha-1 antitrypsin deficient patients for a long time, the more the severity of the disease is, so the more severe the emphysema is, the more severe the airflow obstruction is, the more symptomatic the patients are, and the more likely they are certainly to have exacerbations, and the more likely those exacerbations are to last a longer time. So if anything, you could argue that what this trial has done is it's biased it against getting the result that was achieved. So that's the way I would look at it.
Mm-hmm. So what about the safety of this? I mean, we've had in the alpha-1 community several recombinant inhaled products that have failed because of safety reasons. This seems to be well tolerated. Is that what your impression is from the study? I don't want Rob to answer this.
Well, it certainly doesn't surprise me. We've been giving plasma purified alpha-1 to people with cystic fibrosis for many years, albeit a more intensely inflammatory milieu, without any safety problems. The problems which arose in the past arose from recombinant molecules which have been contaminated for whatever reason. And I think that is not a problem with the plasma purified proteins which you have at the moment. It doesn't mean we cannot use recombinant molecules in the future, but it's just that we have to be extra careful, I think.
Something you want to add?
No, I mean, it was a trial in which I didn't think we saw adverse events above and beyond anything where we got a placebo. It was quite a pleasant trial.
What do you think about that peak in the beginning of the trial of adverse events? In both groups, really.
I'm tempted, and I don't know the data for sure, but I'm tempted to say that was Birmingham. And the reason I'm tempted to say it was Birmingham was that our pharmacy department just happened to release the first dose, which had an expiration date on it that was the day or two before the patient came to see us. Now, that wasn't an issue. It's just they shouldn't have left it on there. So the first thing that happened was that first patient had a bad reaction. And then it gets around. The patients are saying, oh, in Birmingham, they're giving you out-of-date drugs. And it took a little while to get that back. And so I would be very interested if those very early events were just our sight.
Interesting. Go ahead.
Just one further point about the efficacy of plasma purified alpha-1. There is emerging data that glycosylation plays an important role in the anti-inflammatory effects of alpha-1 antitrypsin. So if you look at plasma purified alpha-1, it's very effective at inhibiting interleukin 8, TNF-alpha's actions, LTB4, whereas non-glycosylated alpha-1 has less effect on IL-8 and other inflammatory molecules. So again, that might be an added consequence of having plasma purified alpha-1 as your molecule rather than recombinant, differently glycosylated alpha-1.
Interesting. Well, I can say that, and this may be an action unique to Denver, that when we start patients on IV augmentation therapy, especially patients that have some production to their cough or something like that, that we see a lot more cough and sputum during the first month or so that they're on augmentation therapy. I don't know if it's hydration or it's, just the ability of this to turn off inflammation and allow the body to start to work on the clearing secretions and things like that. I don't know if anyone else has had that experience. Well, come to Denver if you want to clear your secretions.
But I think that's an interesting observation, and we shouldn't just say, "Oh, that's interesting." It's telling us something. Mm-hmm. I would have said, "Well, that's counterintuitive," because a lot of these patients have free elastase activity in their airways, and the one thing that elastase does is it makes you chuck out mucus. So if you inhibit it, you'd expect it to go the other way. The alternative is, in fact, what you're actually doing is opening up airways, so they're now starting to cough it up when they couldn't before. So it's an important signal. I think we need to look at that.
So I wanted to just kind of ask a couple more questions, and then we'll open this up to the general audience. And these are more general questions about, Jan, is this a convincing study to you at this point? Is this something that, if this drug were available, you would start to give it to your patients?
Well, that's a challenging question. At least it's a surprising result, which we all had hoped for. But given the effects seen in IV treatments, yes, it's really surprising. Based on the data, I would say it is really worthwhile continuing exploring what this treatment means over the long term. So, in other words, is the improvement that we tend to see in the results in this specific repeated measurements analysis in the mixed effect model, is that for real and holding over a certain period of years? Yeah? And on top of that...
Sounds like you're saying that some post-marketing studies might be required.
I think that would be worthwhile doing. And then, of course, the other thing that might be intriguing is to see if we can improve the dosing by just looking for other nebulizers which may have an improved deposition efficacy, especially in the small airways, as we learned yesterday and the day before yesterday during sessions here at this meeting, that the small airways is the thing to go for. And I guess also in Alpha1 patients, although that remains to be determined.
I remember accompanying Kamada to go talk to Perry about what was the best delivery device at the time that the studies were being devised. And, that's the problem with getting a delivery device. You're locked into the delivery device that you start with, and the technology improves as time goes on. It's one of the issues, though, in regulatory policy, because you're often approved with the device that you did the study in, which locks you into a device that doesn't necessarily lock the clinician into the device, but locks the company into recommending administering it with a particular device. So the big final question of mine is, here's a study. It is the largest inhaled alpha-1 study that's been done, certainly in terms of a placebo-controlled randomized trial. Everyone wishes that the study had gone on for 5 years instead of 1 year, but we probably wouldn't have a Kamada to do the study if it was going on that long. How do you think this is going to affect the treatment of alpha-1 into the future? Anyone? Go ahead.
I think it opens up a number of potentials that we hadn't got before. So one of the questions would be, how does it interact with intravenous augmentation therapy? Is it complementary to it? Does it replace intravenous augmentation therapy? Or are they both really targeting different parts of the disease condition? And should one or either of them be used during an exacerbation, in addition to the constant use of the other form of therapy? So it opens up a lot of questions to us that we didn't really think we could answer in the past, but now we have, I think, an opportunity to look more closely at these. And I think it tells us an awful lot more about alpha-1 antitrypsin deficiency than we knew heretofore.
Well, of course, sitting in the UK, we haven't got IV therapy like Jan and a couple of the other countries around us. So having a positive study that actually achieves a license is a great step forward. And in places where you haven't got IV therapy, you can ask different questions to the ones that Jerry's just been raising. And I think particularly in this patient group, if we had availability of this drug, I think it would be very important to follow densitometry, which has been previously, often through tough times, accepted as an outcome measure as well. And does this deposition in the airways affect what's going on at more peripheral levels, at alveolar level? And I think that would be a very key question. And if the answer to that was yes, well then why would you start to go back to IV therapy?
Yeah, that will be a major challenge to investigate whether this has an effect on emphysema component in this disease. And
well, you did the CT densitometry. Maybe you could just end by kind of commenting on that.
Well, yes, this means that we now do have the tools, and they're being recognized, and others are trying to improve this tool, which is good. And hopefully, the regulators will now allow us to do those kinds of studies with inhaled product and see what happens. And of course, that should be the other ultimate goal, is not only to improve lung function, but also to halt the progression of emphysema to our patients.
So with that, I would like to open up the questions to the audience. I see a lot of very knowledgeable alpha-1 treaters and researchers. Are you working with a microphone? Good.
Mark Brantley, University of Florida. I wanted to know, going back to the primary outcome variable, have you done any analysis based on season? Because, I mean, this was essentially a one-year study, and we know that exacerbations are more frequent in different seasons. Were you able to, could you take a look at time of enrollment by season and analyze those like by quarters or something like that?
Well, we could.
Did you?
Not yet. We are planning to.
But P is right. We have got that data. Oh, have you got an answer?
Do that yet.
No, but because they were recruited throughout the year. So they would all have had an annual cycle. And the point you make is reasonable to look at. Was it making the winter exacerbation rate different, even though the overall rate was not different?
Do you know whether there was equal enrollment in the seasons of the year? If a high enrolling site came on in a particular season?
It pretty much did. I think we had more difficulty in the winter because, of course, they had to be free of an exacerbation before they could be randomized. And so there was problems sometimes with patients you just get ready to randomize them and you'd have another chest infection. Throw them back.
Problem
3
with
more
frequent exacerbators, trying to get them enrolled when they're not exacerbating.
Well, I can add to that we did do an analysis for those centers who contributed most to the study, which I think was about 70% of the patients, where we looked at the history of the 2 exacerbations that occurred related to the first exacerbation in the study. Yeah? And this was homogeneously distributed along the study conduct, I would say. So there was no difference. It wasn't that the first exacerbation occurred in a patient that had its last exacerbation more than 1 year ago or 3 months ago. This was nicely distributed. So, and on the other hand, in my experience in Western Europe, where the study was conducted, the flu season was in that year, those 2 years, not much different from other seasons. And I guess the flu component here is the most important component.
Keep your hand up.
Hi, Andrew Fine. Just a couple of questions, I guess. One is forward-looking. So given that when the time comes to think about a potential U.S. trial design, this very same group is all likely to be asked for input. How do you think about these results as potentially informative of a regulatory study in the U.S.? And then a second question, I guess more to Dr. McIlvaney's point about the applicability, perhaps, of these results to other airway diseases involving inflammation. And that's, so we've heard recently that Grifols' attempt at administering prolastin in the AATD nebulizer in CF, produced some results that, may not have been what people were hoping to see. I guess, is that simply the derivative of trial design? Is that, different disease? Is it that this might be having a more powerful, for whatever reason, anti-inflammatory effect than other forms of Alpha-1? So just your broad-based thoughts there as well.
Why don't you take the second one?
Yeah, I'll take the second question, if that's okay. So, I was involved in some of the very early trials on aerosolized alpha-1 antitrypsin to people with cystic fibrosis. And the one thing that strikes me about cystic fibrosis is the enormous variability of the neutrophil elastase burden in the CF lung. So it's enormously different. So it goes from 50 micromolar down to 0.5 micromolar. So it's an enormous variability. Secondly, there are a lot of structural damage in the CF lung, a lot of bronchiectasis, a lot of mucus plugging. The disease is characterized by mucus plugging. So a lot of these things are really biased against an aerosol delivery system of alpha-1 antitrypsin being effective in inhibiting neutrophil elastase. Despite that, the data from our own group years ago and from Mel Berger some years after that was that if you give enough, you can inhibit neutrophil elastase. And the holy grail of inhibiting neutrophil elastase in the setting of cystic fibrosis is that it should have an effect on Pseudomonas aeruginosa colonization, and that's a really important area in cystic fibrosis. So that's one of the things you don't have in alpha-1 antitrypsin deficiency. It's slightly different. But the real problem in CF is the amount of neutrophil elastase you have to inhibit, the structural abnormalities there, the prevalence of mucus plugging. So it's not impossible, but it is difficult. And it's all down to delivery devices and perhaps also the use of mucolytic agents or such as recombinant DNAs or whatever beforehand to clear out the airways as much as possible. So I would never give up on using aerosolized alpha-1 in the setting of cystic fibrosis, but we're only now beginning to understand airway inflammation in CF, and for example, you have a lot of glycosaminic glycans in the airways in CF which will inhibit some of your antiprotease factors. And all these things come together in CF which make it qualitatively and quantitatively different than alpha-1 antitrypsin deficiency.
I guess as the American, you want to follow up on that?
I was just going to ask quickly, so in that context, are there then other diseases, pulmonary disease involving inflammation, where neutrophil elastase might play a role more akin to that in alpha-1 deficiency?
Well, non-CF bronchiectasis is a classic one. Again, less neutrophil elastase, less mucus plugging, less inflammation than you do get in cystic fibrosis. And alternatively, starting anti-protease therapies early in cystic fibrosis before you have the structural abnormalities which cause so much of the problems.
So, you know, obviously I don't have an insight, maybe other people in the audience that have a more definitive insight into the U.S. regulatory process, but I haven't found them to be as, at least in terms of alpha-1 drugs, as easily moved from their dependence on primary endpoint. On the other hand, an approval in Europe with good experience in Europe could, of course, push regulatory authorities in other countries, including the U.S., to take a second look or a stronger look at the entire, you know, pattern that's emerged from the study. So we just have to see what happens, and you know, Kamada may be able to comment on that. I really thought I would take a stab at it at the start because they probably can't comment on it.
I guess my question was more along the lines of, you know, going forward, if one assumes that at some point an additional study is likely to be required prior to the FDA approving this, what should that study, in all of your opinion, you know, probably look like from a design perspective?
I would say either before or after a marketing approval, because that's one of the things the FDA has done, is give marketing approval with a commitment to do further studies. So, comments?
Are we just talking about alpha-1 antitrypsin deficiency?
Yes. Yes.
Well, from a personal perspective of trying to watch inflammation over decades, what Jerry has just brought up about cystic fibrosis is the horrible end of inflammation. Once inflammation gets going and you start to develop tissue damage, then it becomes a self-perpetuating problem. And particularly when patients are colonized with Pseudomonas, Pseudomonas throws out its own proteases that inactivate alpha-1 in an incredible way. And I think, as he's intimated, it's difficult to crack it at that point. So I think early disease and early inflammation is where we've got to go. And I know the tendency is not to do that in alpha-1, partly because you don't know what's going to happen next. But if you get people early, you can take your time and see what happens next and pick up the patients who are on the downward slope. And I'd like to see studies done in very early disease and then try and prevent the long-term sequelae and the chronic inflammatory process that we get towards end-stage disease.
Boy, that's a great point. You know, the fact is, this is IV therapy, inhaled therapy is basically a prophylactic disease and a prophylactic therapeutic in alpha-1 antitrypsin deficiency, and you'd sure like to prevent every bit of elastin degradation that you can prevent. And if we had a way of treating patients at the earliest signs of disease, it's only logical that would be the time to start it.
Certainly Rob's suggestion has tremendous appeal. I also find myself going back to an earlier thought, which is perhaps at certain times, so during exacerbation, one might top up the patient with alpha-1 antitrypsin deficiency with inhaled therapy to, in effect, produce the acute phase reactant result that would normally take place in a non-deficient individual. And I'm trying to think of the regulatory or social context for this. Augmentation therapy is very well used in the U.S. There's some reluctance not to use it, so if one is not going to do away with the established intravenous augmentation therapy, perhaps the U.S. question is a top-up relevant, a top-up that arrives via a different route?
Yes.
I'm not sure how well taken forward this recommendation in 2003 was with respect to the guidelines how to treat these patients, but it was recommended to treat patients with an FEV1 in the alpha-1 population between 65 and 35% predicted, right? Which indicates that you have to stop IV treatment in that population once the FEV1 is below.
Trust me, that hasn't been lost on insurance companies in the United States.
So, although I fully agree with Rob's approach to study early on disease, I also would then recommend to study for the FDA what happens if you treat those individuals with an FEV1 below 35% of predicted, and see what that does on both FEV1 and on survival.
Well, there will be a new treatment guideline in the United States soon, we hope, that will hopefully eliminate some of those issues.
I think the reason that this is all highlighted is because that was registry data. That was where the difference in decline in FEV1 was seen. But that's also the time when the decline in FEV1 is its greatest in a group. And so if you're looking for an outcome, what you do is you look for an enriched group. And so if you've got a fast declining group, that's where you'll see your best outcome on that decline data. But you go to the end of the disease, and the emphysema process is progressing just at the same rate, and gas transfer decline is actually greater the more severe the disease than early in the disease. And so just taking data out of context like that can lead to the wrong decisions. And I think that it's also happening in Spain. I think Mark Miravichelis was telling us that he has to struggle a lot now when these patients get their FEV1 down to 30% to continue therapy. They have to stop, yeah.
Next question.
In reviewing the electronic diary, was any differences noted in febrile events or other infections other than the ones that were just reported?
Other infections, there were certainly episodes where antibiotics were given that didn't seem to be appropriate because there was no change in respiratory symptoms. So we're getting back to that data to see, well, what was the indication for that treatment. Febrile events, yes, we've also collected that, but we haven't yet analyzed that as a specific endpoint yet. But we've got that data.
So on the antibiotic usage, you haven't analyzed the usage of one group compared to the other?
Not at this moment in time, but we've got that data. So it is now the character of those exacerbations and how was it treated or not treated, which will be another way of looking at the data. So you're right.
Thank you.
Next question. There's one over there.
Hi, could I just ask a little bit more about the 4-week post active treatment data? You said that had returned to baseline. Is that literally the case, that the means were back at the baseline values for both treatment arms?
Well, all I can say about that is that measured 4 weeks after the last inhalation, there was the mean value of FEV1 returned close to the baseline values, yeah. And I don't know the standard deviation of that particular measurement, but this is, I think, what we measured.
I mean, obviously, that's a question in terms of whether we can say that these data show that the natural history of the condition has been changed, or whether this is more an effect like with tiotropium, that you get an improvement while you're on it and then you're back where you started afterwards, like in, say, uplift.
But I think a 1-year trial for changing the natural history is utopia. So,
was that only in the treated group that you saw that return?
Yeah.
Both groups?
Yeah, in both groups.
So again, one thing that follows from that is there may be an adverse effect of inhaling the placebo. But if the lung function improves when they stop taking it. I mean, I think it's best to say it in this environment rather than later on when the reviewers are reading it.
We should look at that.
I noticed that there was a 50-week follow-on. Did you collect data during that time? Oh. Any other questions? Come on, the tough ones. Don't hesitate. These guys can take it. Well, if there are no other questions, do you want to say something at the end, or shall I simply thank everyone? I will do that then. I especially want to thank the panelists for their expert opinion and for allowing me to sit in the middle of them and be honored in the glow of their expertise. And thank you all for coming to this and making this a successful presentation and question and answer period. And thank you, Kamada, for being such generous hosts.